In this experiment, we will study the structure of a pig heart. Wash a pig heart thoroughly with tap water. Use a pair of scissors or a scalpel to remove any fatty tissue that surrounds the blood vessels of the heart. Identify the dorsal and ventral sides of the heart. Identify the left and right sides of the heart. Identify the left atrium, the right atrium, the left ventricle, and the right ventricle. Press on the heart gently. Feel the thickness of the ventricles. Identify the aorta, pulmonary artery, vena cava, and pulmonary vein. Identify the coronary blood vessels running on the surface of the heart. Draw a labeled diagram of the external structure of the heart. Use a water hose to run water slowly into the aorta and pulmonary artery, as well as the vena cava and pulmonary vein. Note what happens. Dissect the pig heart with the ventral side facing upwards. Cut the ventral halves of the ventricles from the bottom. First, cut along the left side from the bottom, and finally along the right side. Cut and remove the ventral halves of the atria to show the bicuspid and tricuspid valves. Cut open the base of the pulmonary artery to show the semilunar valves inside. Cut open the base of the aorta to show the semilunar valves inside. Study the internal structure of the dissected heart. Compare the walls of the atria and the ventricles. Compare the walls of the left ventricle and the right ventricle. Identify the tricuspid valve and bicuspid valve located between the atria and the ventricles. Identify the heart tendons that hold the valves. Identify the semilunar valves at the base of the aorta and the pulmonary artery. Draw a labeled diagram of the internal structure of the dissected heart.